I'm going to be talking today about cereal diseases in the southern region and focusing more on the low and medium rainfall zones. And if I can get, there we go. So just a summary of what we've seen so far in 2021. A lot of regions experienced quite late opening rains. And this meant that crops were sown in either late May or June into quite cold soils. Therefore, crops were quite slow to start, and despite having a higher than average rainfall, they've been a little bit slower in development. We've had some reports of low levels of disease, particularly in the medium rainfall zone, but a lot of growers are saying that it's not moving up the plant too much and it seems to be sitting below in the bottom area of the plant. So a bit of an overview of what I'm going to go through today. So this is a summary of all of the fungicide resistance that's currently known in Australia. We're going to touch on the barley net form net blotch groups 3 and 7, the wheat powdery mildew in group 3 and 11. And then I'll touch a little bit on the pathology of wheat Septoria tritici blotch and then hand over to Nick to expand more on the fungicide resistance in that. So net form net blotch, the way that this disease moves is predominantly stubble borne. So the disease survives on the stubble over summer and then it produces spores which move onto the young plants. This can also come from seed, but this is not probably the predominant source of infection. It's reliant on having a very high level of disease in the crop that the seed was sourced from. And it's a much slower moving start to the disease than the stubble borne, which can really affect right across the region. As those lesions develop, we get the conidia forming and they can move up the plant and from paddock to paddock and longer ranges. This disease has a relatively short latency period, so we usually see from kind of 7 to 10 days, but it can be shorter if the disease conditions are optimal. It really likes high humidity and it does like warmer temperatures as well. So what are our risk factors for net form net blotch? Growing susceptible varieties with very little diversity in the region, growing barley on barley with stubble retention, early sowing, and warm, humid weather. And we mentioned that the short latency period will allow the disease to move quite quickly when these conditions are met. So in terms of fungicide resistance, I'm sure most of you will have heard me touch on the SDHI situation that developed on the Yorke Peninsula in 2019. Basically, the year before was a paddock of Spartacus. It had a full rate of Sustiva. The disease developed pretty well, and then that same paddock was sown to Spartacus with a full rate of Sustiva in 2019, and the stubble was retained from that previous paddock. So the disease developed quite quickly, and you can see in the picture here that we've got quite a lot of disease going up the plant. This was also a similar situation to many other paddocks kind of in that region. So we sent some samples off to Fran and Wes at CCDM, and they confirmed that we did have the SDHI resistance in net form net blotch there. We then conducted a survey in conjunction with the CCDM right across the Yorke Peninsula. Basically, what you can see here in this map is that blue means sensitive and that the pathogen is still responding to the SDHI. Orange is reduced sensitivity and red is resistance. So as we move down the Yorke Peninsula, you can see a lot more of that reduced sensitivity and resistance down there. Moving into last year, we continued to do more sampling as we spread the word about what had happened on the Yorke Peninsula in 2019. And you can see a pocket up here on the Western Eyre Peninsula where we've got quite a few highly resistant paddocks, some reduced sensitivity in the middle of the Eyre Peninsula. The upper north, these greens, these are indicating sensitivity, so the fungicide's still working. The Yorke Peninsula as expected. And over here in the mid-north at Avon, we've got another case of highly resistant. Down in the southeast, we had a sample of reduced sensitivity down at Carby-Bowlat. Moving on to this year, and there was a report from Victoria of netform net blotch developing in Spartacus that had been treated with Sestiva near Oyen, and that's now been confirmed to have the resistance as well. So that is now confirmed to be in Victoria as well as South Australia. Whilst we were conducting the survey for the SDHIs, the DMIs were also tested, and as you can see from this map, there's a lot of orange and red across the Yorke Peninsula. So this is Tebuconazole as an indicator for the DMIs. So what lessons do we need to know when we're managing fungicide resistance for netform net blotch? And I'm going to bang on a lot about this today, but integrated disease management strategies. So it's really about reducing the severity of the disease. The primary way that we can do that is using varieties that have resistance to that pathogen. We really want to avoid very susceptible and susceptible varieties. Crop rotation. So if you know netform net blotch is a big issue in your area, then try and reduce barley on barley and decrease the frequency between your barley paddocks. Stubble management, we know that there's some great benefits to retaining stubble, but I think it can be a big driver for disease. So it's worth thinking about, is there something that you can do to reduce the residue that you're planting into, whether that be late grazing or burning, or another option, but it's something to consider. In terms of fungicides, we need to be rotating our modes of action and avoid using the same active twice in one season. If you know you have fungicide resistance for a particular fungicide and pathogen in your area, then avoid using that in that year. Powdery mildew of wheat. In terms of the life cycle, this is another one that is stubble borne. But it can come off volunteer plants as well. Basically, young plants get infected directly from the stubble or from volunteers, and the spores that are produced from those lesions can then be spread through the wind. They then move up the plant as the season goes on, and when we see them in the head, then this is where we start getting into yield loss territory for powdery mildew. This one is another one that has quite a short latency period and the disease can develop quite quickly. Rain splash, the rain can actually wash some of the spores off and stop them from being aerosolized. It's more the humidity that's the main driver of disease with the powdery mildew. So similar risk factors with the powdery mildew to the barley netform net blotch. So growing susceptible varieties with little diversity in the region, growing wheat on wheat with stubble retention systems, early sowing and humid conditions. So in terms of fungicide resistance in the southern region, so we've got resistance to all group 11s, the strobes in New South Wales, SA, Tas and Vic. The group 3 fungicides, we have resistance in propiconazole and tebuconazole in New South Wales and Vic, and lab detections of group 3s, which is a gateway mutation, so more of an early warning sign in New South Wales, SA, Tas and Vic. In terms of managing this fungicide resistance, once again, integrated disease management strategies are your first go-to. The variety selection, once again, we see a lot of susceptible and very susceptible varieties for wheat powdery mildew being grown. If you know it's an issue in your area, try and avoid them and find a variety with better resistance. Crop rotations, like the barley, we see wheat in very close rotation in areas where this is an issue. Stubble management again. With wheat powdery mildew, controlling the infection early seems to be a big factor in whether or not you get on top of the disease during the season. So if there's a seed or in-furrow treatment available, then that's a good option as well. And once again, with the fungicides, rotating our modes of action and avoid using the same active twice in one season. Septoria tritici blotch, this is another stubble-borne disease. You can see this is quite a theme, and certainly the way that our stubble retention systems work drive what diseases we see in our system. So the ascospores come directly off the stubble, and these can go really long range in carried by the wind, and affects young seedlings. As lesions develop on those seedlings, we get these fruiting structures that produce conidia. They're then spread up the plant via rain splash during the season. So having rain is probably one of the main drivers here. Septoria tritici blotch is a little bit different in its latency period. It's not as fast moving. It can be more like 14 to 42 days or 250-degree growing days. And it really varies based on temperature. So the warmer the weather, the faster it can move, and the other conducive conditions that go with it as well. And I'll leave a little bit more for Septoria tritici blotch for Nick in a minute. So growing susceptible varieties, again, stubble retention and close rotations, early sowing, and frequent rain events. So thinking about 2021 and where we're at now and what we're expecting to happen, a lot of areas have received quite a lot of winter rainfall. Even though the crops have been moving slowly, this is setting up for larger canopies in spring. There have been some predictions to say that spring rainfall could be higher than average as well. And the low levels of disease, these could move quite fast once we start to get conducive conditions and warmer temperatures. These warmer temperatures can shorten the latency periods for many of the diseases. So now's a good time to get out and monitor your paddocks and consider what your strategy is for the rest of the season. Fungicide resistance in practice. So we know that there's more fungicide resistance out there. We're hearing a lot about it, but how does that actually play out in the paddock? It's not going to mean every year that you put that fungicide out that's resistant and it's not going to work. We've got very diverse pathogen populations, and the resistance can be a proportion of that population. So the maps that I showed from the York Peninsula, these are an example of the population within one paddock, and you can see that there's quite a lot of diversity. One kind of pathotype doesn't dominate an entire paddock. So we can have very low proportions of that resistance in the population. Really, it's the conducive conditions that we'll select for this resistance and the management strategies that we're using. We want to be kind of selecting away from that and not encouraging this disease, fungicide resistance to proliferate. And pathogen fitness is a factor as well. So the mutations don't necessarily mean that it is the most aggressive pathotype as well. But once again, selecting for this will give it the capability to develop further. So it's just important to remember that we're not necessarily going to see this every season, but it's out there and we need to be thinking longer term about how we're managing our paddocks. Fungicide resistance management, more generally, I think I've touched on this for each of the diseases, but it's not just about our fungicide choices, it's these integrated disease management strategies, and that starts with variety selection. So where possible, we should be selecting varieties that have resistance and trying to avoid susceptible and very susceptible varieties. Supporting our variety and fungicide choices with crop rotations, management of green bridges, and stubble management. And then when it comes to fungicides, we need to rotate our modes of action, use mixtures of modes of action when they're available, and stay within our label rates. I've talked quite a lot about variety resistance. You can find all of these in the Cereal Variety Disease Guide, and there's a link to that here. We update that every year. And Bridget mentioned the Fungicide Resistance Management Guide at the beginning of the talk. That's a great resource for more information, so check that out. Thank you very much.
Following on from what Tara has talked to you about, I'd like to concentrate on just one of the more recent findings with regards to fungicide resistance, and it concerns Septoria tritici blotch. And I'd just really like to reiterate a couple of things. So Bridget, next slide. The first is that one of the things that's dominating the high rainfall zone, both currently in wheat and in previous seasons, has been Septoria tritici blotch. And I really want to emphasize just how difficult this disease can be to control because of something that Tara's already mentioned, which is the latency period. Because this long latency period, this long time for Septoria tritici to develop within the leaf, actually is one of the primary issues that disguises the disease as it advances. And in fact, you can actually very often find at this time of year, just as the soil's warm, that the crop appears to grow away from it. But because it's a wet weather disease, if there is Septoria tritici in the base of the canopy, very often the advance of the disease in the newer leaf tissue is disguised. And if you go next, Bridget. One of the actual aspects when you look at this is that you can actually develop and a portion of a leaf, so 2 clean leaves and a portion of a leaf as well, thermal time in the same time as the disease expresses itself. And so when you look quite often at a crop at flag leaf, it can very often actually potentially have the clean leaves that you're looking at, the 2 top canopy leaves, can already be infected because of the long thermal time it takes for this disease to express itself. But one other point, and that is, as Tara said, as the season warms, so this disease will express itself far quicker than it does in winter. And typically, once you get into that main spring period, we'd be looking at about 21 days, but these latency periods are covered by thermal time, not calendar date. Okay, thanks, Bridget. So what they discovered, the team at CCDM, looking at some samples that came from our research site down in Millicent, I want to take you through what they've actually found in the Septoria tritici blotch pathogen. I want to talk to you about the fungicides that potentially affects, why it's important, and then how we can adjust our management. So first of all, let's see what they've actually discovered. We have a group of fungicides that we call the group 11 QOIs. They're commonly called the strobilurins, and in broadacre cereals, we principally use 2 active ingredients in this group of fungicides. One is isoxystrobin, found in the Amistar type products, and one is pyraclostrobin that we find mixed with other products in, say, Opera. Now, what's been discovered in Southeast South Australia at this stage, and we don't know how widespread it is or what proportion of the population is carrying this mutation. But what we do know is that this mutation is actually just a single step that turns Septoria tritici from being susceptible to the strobilurins not working to control them. That mutation appears from other parts of the world, Northwest Europe, New Zealand, to be fit in the environment. And this is something, again, that Tara mentioned. You can get mutations, but are they fit enough to survive in the population? In Northwest Europe and in New Zealand, they have been. And if the same pattern occurs, what it means is that this pathogen mutation will quickly advance in the population if we continue to select for it with using strobilurins. And so in Europe and in New Zealand, it took approximately 3 years for it to go from a situation where strobilurins worked to control Septoria tritici to when they were ineffective against it in the field. So one of the things that we need to be aware of if we're setting out to control Septoria tritici this spring is to make sure that we know if the products we're using, by discussing it with our advisor, actually contain the strobilurins. And I'll come back to the fact that we may well still be able to use them this spring because we don't know how widespread this mutation is. And if only 10% of the population contains that mutation, the other 90% may well be very susceptible to strobilurins. And it is important to note that the strobilurins can control a wide range of diseases, not just Septoria tritici blotch. This aspect of strobilurin resistance actually comes on top of something that's been identified previously by New South Wales DPI, and that is that the triazole group of fungicides, so next slide, Bridget. The triazole group of fungicides, this is the products that we commonly use, the tilts, the folicures, or the tebuconazoles, the propiconazoles, the prothioconazoles, the epoxyconazoles. We already knew that their performance could well be affected against Septoria tritici in them not perhaps performing at the rate that they might have, say, 5 or 10 years ago. And we know that some of those group 3 DMIs are actually perhaps more affected than others. A good example of this is tebuconazole, for example, that a lot of people might use as a good wheat fungicide that's good on rust, but its activity is probably not there, and it doesn't currently carry a label recommendation, but people may well be looking at that for some level of cheap control, and it's not as efficacious as some of the other DMIs. We've been working on looking at fungicides for the management of Septoria tritici in the very region that this resistance to QOIs has been discovered. We've been looking at that particularly in 2018 and 2019, and I'm going to show you some of our data from 2018 first. So next slide. So this gives you some examples of efficacy of different fungicides at their highest label rate, a 50% dose, and then a 25% dose of that full label rate. And what I'm trying to indicate here is the performance of straight DMIs, and in this case, tebuconazole, ciproconazole, which is the DMI active in Amistar Extra, Opus, which is present in products such as Radial with the strobilurins, Taser Expert, for example, and Prosaro. Now, Prosaro is a mix of prothioconazole and tebuconazole, so half of it we know is perhaps not that effective. But what you can see when we looked at them in 2018 is that they may not be giving us 100% control, but particularly the ciproconazole and the epoxyconazole look to be performing well. You can also there see the performance of mixtures of DMI the triazoles, and the strobilurins that have now, in this last month, been shown to be affected by this new pathogen mutation. You can see there that they're working through their rate range, and azoxystrobin, for example, and radial were extremely effective. Then two other products, one containing an SDHI mixed with a DMI in aviator extract. So we're not short of fungicides in 2018. If we move to 2019 to look at the performance, in this case against Septoria tritici and leaf rust, and the Septoria tritici is in the dark green color. And what you can see is that there are different timings of fungicides here, either a single spray at growth stage 31, a single spray at flag leaf, and then a 2-spray approach. And you've got a range of products there that again look to be working effectively. And all I'd point out is that there we've used the mixture products at the lower end of their rate range. So the radials, the aviator X-Pros. We've used a lattice at its full rate, which is a mixture of the DMI and the SDHI. But I think what I'm trying to show you, generally speaking, is to keep Septoria tritici and leaf rust off of the flag leaf in that Millicent region. You needed to look at a 2-spray approach, particularly with emphasis on the flag leaf. What you can see there is that actually, again, we had products that were giving a reasonable level of control. In fact, as you can see, the straight epoxyconazole there actually performing quite strongly. The point I'm trying to make here is that when we talk about resistance, this isn't Armageddon. It doesn't mean that our fungicides don't work, but it does mean that we need to be aware of what potentially the consequences of this resistance might be. If you look at the yield effect, and this was on the very susceptible Septoria variety revenue, but if you look at the next slide, you see the effects of those disease control measures here measured in terms of yield. And you can see that there's the most perfect correlation with the kind of disease levels that you had on the flag leaf. Again, I really wish to point out that the fungicides that we're commonly using giving us tremendous responses in the presence of Septoria tritici early and leaf rust late. Clearly, we can't differentiate there between which part of the yield response is exactly leaf rust, but nonetheless, you can see 2-spray measures giving very good control. The coded product on the left isn't yet available to us, but that will be the product that contains a new DMI mixed with fluxapyroxad, and that product will be, when it's commercialized, called Revistar. So that's the FAR F119. Okay, so where does that leave us? If we move to really the situation this spring, the first thing to emphasize is this disease disguises its advance with a long latent phase. So if, as the spring warms up and the crop moves through its stem elongation phase to flag leaf, and you look at the crop and the top of the crop looks clean, it may well be clean if you've had dry weather for all of that stem elongation. But if it's been peppered, as Tara was saying, with frequent wet spells, it could well be that any Septoria in the base has moved on to those newer leaves. This spring, particularly in the southeast of South Australia, we have to be aware that the strobilurins might be impacted by this new single-step mutation. In 2 or 3 years' time, if this mutation works as it has in other parts of the world, it may well mean that the strobilurins, we will not be able to use them as a reliable control measure for this pathogen. However, at this stage, we don't know how widespread it is outside of Millicent, and we don't know what proportion of the population is affected. So if you're using a QOI for other diseases such as RUS, then I think the advice for this spring is that whatever you're mixing that strobilurin with, and nobody applies strobilurins on their own in wheat for the control of these diseases, then make sure that the partner, the group 3 partner DMI that you're mixing with your strobilurins is at a robust rate. So if you haven't put much strobilurin into the mixture and you're using a lower rate, what I would be tempted to do is to top your DMI rate up in order to make sure that the DMI alone is giving you good control of the Septoria, just in case. More information on the AFREN website. We've also done a YouTube clip to assist you with a little bit more information around what's showing up at the moment with regards to this disease. But I think, like I say, this spring, we're a little bit of an unknown as to how widespread this problem is. So it's not discarding the strobilurins in any way, it's just being aware that it could be out there. And where you've used them, monitor the effect with your advisor. Many thanks, Bridget.
Thank you very much. Nick, just a question. If I'm looking in my crop and I can't find Septoria yet at this stage, should I still consider spraying?
I think this is one disease, Bridget, and there's a lot of later sown crops, as you were saying, in South Australia early. I think this is one disease that if, as you come up to stem elongation, you can't find this disease, or it's there at incredibly low levels in the lower part of the canopy, this is one disease that we know probably won't infect our crop from now on. So unlike rust, it's not one of those diseases that will come over the fence at us. So if you haven't got it at this stage, I don't think you're needing to take that into account when it comes to your control measures for this spring. Sure, rusts and such like powdery mildew, but if you haven't got it in the crop by now, then I think you're unlikely to see this just appear like a rust would appear.
So, yeah, look, my talk is really going to be more of a seasonal update and then how we're going to make fungicide decisions in the next couple of weeks. So, firstly, I just want to touch base on where we're at and how we've got here this year. So, with blackleg now, we're really sort of talking about the disease having really 3 key infection periods and almost sort of handling them as actually totally independent diseases. So, our blackleg, obviously, like Tara was saying, with a lot of other stubble-borne diseases, is surviving on canola stubble from last year's crops and producing sexual wind-blown spores. Now, when we see those land on our seedlings, we see that the lesions cause on our cotyledons and our leaves, and the pathogen grows within the vascular tissue down to the crown, and we get crown cankers or partial infection, which causes yield loss. That's sort of like the traditional blackleg, which everyone understands pretty well. And then more in recent years, we've seen exactly the same system happening. So spores from stubble, but this time landing directly on our plants as they're going reproductive in late winter. And then we're getting what we're calling upper canopy blackleg infection. The third type is exactly the same process occurring again with very late rainfall, and we're seeing infection directly onto the pods, which we're calling pod infection. But today, really what we're going to concentrate on now is this upper canopy infection. Like the spray applications are obviously too late to protect our seedlings and crown canker, but we're right now coming into this window for upper canopy. First thing I want to look at is we're talking about different seasonal effects, and with blackleg and when these 3 different forms of disease, as I said, we're almost targeting them as different diseases, and the seasons we've had the last couple of years have been very dramatically different. So in 2020, we had like the perfect start to the season. We had obviously a very wet spring, and then we had dry winter, and then a wet autumn, dry winter, then wet spring. So it's the perfect growing season. But what that meant is that we had this opportunity to sow our crops really early. Whereas in 2020, it was obviously been a very different situation. In 2020, we had this really late break to the season, and therefore our crops are obviously sown a lot earlier, and we've had this really wet winter. And what this means is, if you look at the 2 years here, last year we had our sowing window, and we really got our crops in early in that April scenario. And if you look at here, here's our spore release. And what I really look at this is where our blackleg infection is occurring. And here we are. So in 2020, we sowed early, which means we got our crops in good warm conditions, and we basically got the crops through that seedling stage before the disease came in. So we had really low levels of crown canker. But then because we sowed so early, our crops were flowering in this late July, August period last year, which really coincided with the period of the year where we get bad blackleg. So we had an upper canopy infection year. That was a really nice scenario for us. We just, no crown canker, but we had to control our upper canopy. In 2021, we've had, I would consider across southern Australia, a much, much bigger window. So in some regions, we still got rain in April and people have sown. And in some regions, we didn't get that well into June. And I've put here sowing germination because obviously a lot of people sowed early, but we still didn't germinate our crops until well into June. And in fact, we've seen a lot of paddocks where we've had 3 or 4 different germinations within the same paddock, depending on different rainfall events. So what this has meant is that we've got some crops which were sown later into winter. They've germinated into this high infection period. So we're back into that more traditional crown canker. But then we've also got a lot of crops which were still sown and germinated early and have put us into this window for upper canopy. So we've almost had both scenarios happening this year. And really, I guess from a disease perspective, what have we seen? I think we've seen fantastic effect with fungicides this year. So I expected to see a lot of crown canker, but the reality was with our new SDHI seed treatments, that early infection has been pretty low. And we've actually, I think, controlled combination of good genetics and fungicide. We've actually controlled our seedling and crown canker pretty well. But as we've come into late July, early August, we've seen this massive outbreak of blackleg. And if our crops were vegetative in that stage, we've seen lots of leaf lesions. And if our crops were flowering that stage, we've seen a lot of flower infection. In fact, we've seen a few crops at the moment where nearly all of those early flowers have been taken off with blackleg. So this is just a really response, I guess, of the amount of rainfall days we had. I just did a quick calculation for Horsham. In the last 45 days, we've had about 32 days where it's rained. So each of those rainfall days is another infection period. So they're just piling up. Every couple of days, we're seeing another infection period, and there's really been no escape for the crop. So we're now beginning of August. We've seen all this happening. So what fungicide strategy should I take? The first thing, and I said here before, we're really talking about upper canopy now. It's too late to control crown canker, and I think our genetics and that have done a really good job anyway, and our seed treatments. But the first thing I want to look at, whether you're making that decision to spray for upper canopy, is have you got leaf lesions? If you haven't got leaf lesions, it's really a low risk. And that means that the genetic resistance in your cultivar is standing up. It's a major gene resistance, and the blackleg's not getting in, and the plant is really immune to blackleg right the way through its life cycle, through its growing period, or it means there's just no disease there. And that would be a very rare situation this year because I'm certainly getting reports that there's been high levels of blackleg right across. Going the wrong way. So the other thing is, as we were talking before, really know what your growth stage is right now. So obviously go out into your paddock. If you were sown early and your crop is now flowering, and certainly if you're seeing little bits of infection, that puts you in a high risk situation. There's probably a lot more crops that look like this, which are just coming up to flower now, but have got disease. So it puts you in more of a moderate risk. And if your crops are still well vegetative and they're not going to flower until late August, early September, that puts you into a much lower risk. So your actual growth stage is really, really critically important. And the reason for that is because we know that the timing of flowering really then affects how much upper canopy we get. And the reason for this is simply that the pathogen, it's the same, works, we think now, as the same as at the crown, that you need the infection to occur, and then it needs time to actually grow into the vascular tissue and cause the necrosis within the vascular tissue to cause the yield loss. So we now think that a lot of the flower infections and external lesions that you see are really quite superficial. It's really this type of disease which is important. So in this scenario here, you've got the same variety sown in the same paddock, obviously under the same environment. One's been sown early, and the early infection has allowed the disease enough time to get in and cause that damage and major yield loss, whereas the late flowering one, by the time harvest comes around, the disease really hasn't enough time to cause that yield loss. So flower timing is really critical. Now, in a normal year, we certainly see that once we get to mid-August, the threat really reduces from that aerial or upper canopy blackleg. So if your crop commences flowering late July, early August, that puts us right in that susceptibility window where we get really good returns from fungicides. If it's more flowering later August, the return for fungicides drops off very, very quickly. The caveat I want to put on that this year is this is the forecast I downloaded today from the Bureau, came out on the 21st of August, and this is probability of above average rainfall for the next 3 months. And you can see, Eastern Australia, the Bureau is predicting a very high probability now, sort of up towards 80% of above average rainfall. And I guess the concern that we have now is that with obviously above average rainfall, maybe cooler conditions, it's extending our growing season for another few weeks. So maybe that critical cutoff period for upper canopy, which we would normally say is around mid-August, might come out a little bit further again. So we're a little bit cautious on saying that if you do get to mid-August, that threat is reducing quickly. But I think the trend still stays the same. But we need a little bit more data with more years like this before we get really confident with that. So I guess the things we've got now, we've got the presence of leaf lesions, we've got our timing to flowering, and the other one we've now found the last couple of years is it's actually the targeted harvesting date. So this sort of goes hand in foot with the, or hand in glove with the flower timing. So if you've got a short period between infection and then harvest, the pathogen doesn't have much time to get in and cause that vascular damage. Whereas if you've got a medium or a longer period, that's really where the yield loss is occurring. So if you've got a later flowering crop and you're in a more mallee environment, the disease probably doesn't have enough time to cause all that level of disease and damage. Whereas if you're in a longer growing season, the disease has got a lot more time. So just sort of running over the points then for this upper canopy stage, things that I would consider. First is sclerotinia threat. If you're going to apply a fungicide at 30% bloom, you obviously also control sclerotinia. Unfortunately, there are no management plans or apps or anything out for upper canopy at the moment, but they are being worked on as soon as we've got enough data. The first thing I'd look at, leaf lesions. No leaf lesions means no upper canopy. That's pretty simple. The more leaf lesions means it's more conducive conditions. The date of first flower is really critical. And then time of infection to harvest. So that time of first flower to harvest, the longer that period is, probably the higher risk. We've got genetic resistance, which we spoke about. There's a good indicator of leaf lesions. And then probably the main driver this year, talking to a lot of agronomists, is this price of the grain. So obviously fungicides are an economic decision, and the higher the price of the grain and the higher the yield, it's obviously much easier to get a return on that investment. So just sort of finishing up here with upper canopy, one of the things is to come back at the end of the year, obviously, and say, did I get good control or not? Or did I actually have a blackleg upper canopy issue? And one of our issues has been that the way the blackleg expresses itself at the end of the year has been incredibly variable. So we keep changing our protocols and our experiments for how to measure it. But there's a couple of ones I think have actually come out more reliable the last couple of years. The first one is I'm just calling darkened branches. So you can just go through your crop and look for these darkened branches right at windrowing stage, or even a little bit after windrowing if there's a few plants left around the place, and you can see they just go completely brown, and that's just a result of them prematurely dying off. So it happens very, very late. You won't see it, like I said, until that windrowing growth stage. But once you get your eye in, you'll pick these up really, really clearly. And the nice thing with this is a much better indicator for actual yield loss. Rather than looking for flower infection and external lesions or whatever, this browning is a much higher correlation with yield loss. And then conversely, the other thing is to get a pair of secateurs and just to grab the tops of the plants and just start cutting them across and looking for that, again, that internal infection, that blackened pith, because again, that's a much better indicator for actual being a real issue. From a fungicide resistance perspective, which I guess is what we're here for today, is our current recommendations for blackleg are really looking at now the blackleg as two separate modes, I guess, infection points for our crown canker and our upper canopy infection. So if you're controlling crown canker, and you'll do that through seed treatments, flutriafol and your fertilizer and that early 4 to 8 leaf, that's one consideration. And therefore we think if you're going to put a seed treatment on and then come back with an earlier foliar, that's the point where you want to rotate your different modes of action. However, we now consider our upper canopy infection to be a separate infection. So therefore, it's possibly safe to use the same fungicide as the seedling foliar because they're totally separate. So if you think about that, if you've used a group 7 as your seed treatment, don't use a group 7, come back as an early foliar. But if you have used a group 7 as your seed treatment, it's probably a lot safer to come back then at that 30% bloom stage. And just making the point, I think like Nick did, that there are quite a few different groups on the market or about to come on the market at the moment. And last point is you can go onto the Crop Life website, and in that there's a section on blackleg, and it gives you all your different management. So if you use no fungicide as a seed dressing, and then use a group 3 early foliar, and then what are your options, etc., for upper canopy in the later of the year. So you can run through all of those different scenarios and pick which one that is going to suit your crop. So with that, I will leave it back for questions.
Excellent. Thank you, Steve. I guess the real key with canola is looking at the prices, and at $800 a tonne. I guess the question is, should I be spraying if I'm seeing blackleg now, even though it might not be flowering? So just wondering what your thoughts are on that.
Yeah, look, it's a difficult window right now. So in previous years, I would say, look, if you're not flowering by mid-August, then no, the chance of getting an economic return are quite small. However, this year, just with the amount of blackleg we've seen over the last couple of weeks, which, like I said, was a result of that amazing infection period we had through late June into July. So we've got a lot of disease, and we've got these really high prices, which changes the economics dramatically. So it's going to be very easy to make a economic return this year. So look, I'm not telling people not to put it on, but just to follow those rules of thumb. Go out, check, have you got blackleg lesions? Have you got new fresh lesions on newer leaves on the first flowers? And certainly if you see that, I would be putting a fungicide on. And with the wet spring, extended growing season, that does increase the probability of an economic return as well.
Great, thank you. And that, I guess my next question really was around what should I do if I suspect there's some fungicide resistance? I'm not sure if Nick or Tara, if you wanted to tackle that one, or Steve. If I'm seeing something I think could be resistance, what do I do?
Go, Tara.
You're more than welcome to get in contact with us here at SARDI and chat to us on the phone or via email about what you're seeing. If you'd like to send samples across for fungicide resistance testing, then we're currently collating samples of netform, net blotch, and Septoria triticide blotch, as well as powdery mildew to send over. So more than happy to arrange that for you.
It's the same for Southern Victoria or Tasmania. If people are wanting to forward samples, I think, as with all these things, make sure that you have done some checks. There's no other suspicious circumstances in terms of application problems that you might have had that might explain why something hasn't worked. But I think just with where we are, particularly with Septoria tritici for this spring, we're interested in anybody who may feel that the products aren't performing like they have been previously.
For blackleg, we're a little bit different in that we really monitor the actual stubble itself and get the stubble to fire blackleg spores onto canola plants treated with fungicide, and then see if the disease develops. So we'll be putting out calls at the end of the year for people to collect stubble after harvest and send that stubble to us. And then come next June, we'll release those spores and test the stubble that way.